So I'll be presenting on kinases in cancer, OLIG2 and Finn kinases tumorigenic potential. Glioblastoma multiforme, or GBM, is a deadly brain cancer that affects up to 200,000 people every year. People who get this diagnosis usually only have 14 months left to live, even after significant chemotherapy, radiation, and surgical treatments. What makes this tumor so aggressive, we ask? Research from the past decade or so suggests that there are a small population of cells in GBM and in other kinds of cancers that are responsible for its aggressiveness. I will be calling them rat cells, or resistant aggressive tumor cells. As you can see here, even after the tumor is hit with radiation and chemotherapy, the rat cells remain and continue to grow and spread. The lab that I work at focuses on trying to find alternative methods besides the traditional radiation and chemotherapy treatments to target these rat cells. And to do that, we need to look at the molecular pathways that are going on within these cells. So, in GBM, OLIG2 has been found to be crucial to these rat cell survival. If we use a genetic trick to remove OLIG2 from these rat cells, we see that they die and there's no more tumor growth. Why can't we just target OLIG2 directly though? This is because no drugs currently exist to actually target OLIG2 because of structural and functional reasons. As a result, we will look to an alternative way to target OLIG2 in these rat cells, and basically I'll be looking at one of OLIG2's co-regulators, or friends, one of its partners in crime. Finn kinase is this friend that I'm talking about. So Finn is also another protein shown to be important in many cancers, and drugs to target it have already begun to be developed. In addition, specifically, Finn is thought to be responsible for the invasive property of cells, for their ability to spread into surrounding tissues and to migrate in that way. So, what is the hypothesis that I have for the interaction between Finn and OLIG2 within rat cells? Since Finn is a kinase, which is a specific kind of protein that adds, let's call them P tags to other proteins, and OLIG2 has sites that can actually receive these P tags, I hypothesize that Finn might be adding P tags to OLIG2 and thus changing its function within the cells. We can think of this as Finn being a bee and OLIG2 a flower. Finn dropping off pollen on the flower and thus changing its function, its overall ability to grow and spread. So if I look into this a little more closely, there are 3 sites on OLIG2 that can possibly receive these P tags, or pollen molecules. And these are tyrosine amino acids present on the protein. So these 3 potential pollen receivers I'm interested in looking at because as of now, I don't actually know if this pollen-flower interaction exists. And these 3 sites are the only possible places where Finn kinase, the bee, can drop off its pollen. My research question is, does Finn kinase actually phosphorylate OLIG2? And if so, at which tyrosine drop-off site does it add these P tags to? And overall, what does this phosphorylation or P tagging event do to OLIG2's overall function? So, since this interaction cannot be directly observed, just because these molecules are so small, the only way I can kind of test it is to defunct these sites where the pollen would usually be dropped off. So I'm going to want to induce a way to make OLIG2's tyrosine sites unavailable for the pollen to be dropped off at. The only way to do this is I cannot change the OLIG2 protein directly. I have to go back to the genetic code that codes for the protein. In other words, if I mutate the DNA at places that code for OLIG2's P-tag drop-off sites, the tyrosines, I can make a defunct version of OLIG2 without tyrosines and thus test its interaction with the fin kinase B. So here is a schematic of what my overall experiment plans to be like. So I want to have a control cell where I have normal OLIG2 with its P-tag sites available. And then fin kinase will come in and it'll be able to add these P's to the OLIG2, and OLIG2 will be able to do its normal function with its P-tags on within the rat cell. In the mutant OLIG2s that I'll compare with the control cells, however, fin kinase will come in, it'll try to add its P-tags, but these sites where OLIG2 usually receives the P-tags are unavailable, and they won't be able to bind. So this is the proposed experiment that I want to conduct to assess fin and OLIG2's interaction. This so that I can see what their interaction actually does within resistant aggressive tumor cells, and then how these resistant aggressive tumor cells then function in the brain as a whole, and specific in tumor. So what I expect, since fin has been shown to allow cells to spread more, invade the surrounding tissues at a higher rate than normal, I expect reduced invasion when this whole mechanism is kind of defunct. In order to do this, as I mentioned a little while ago, I must go back to the DNA that codes for OLIG2. So I used a mutagenesis method called the quick change multi-method to actually mutate the sites on OLIG2 that code for the P-tag drop-off sites. I put the DNA into a thermal cycler, which basically mutates the DNA through various temperature cycles, then digested any unmutated DNA, and finally collected the DNA that I had supposedly made, the supposed mutant OLIG2 DNA. What I actually found was that this procedure didn't work very well with the OLIG2 DNA that I put in, for different reasons though with every trial. In the first trial, here's the DNA chromatogram that shows that's trying to sequence the DNA that I put in, all to see if I actually mutated the OLIG2. And in this process, you can see that the graph is extremely noisy and can't really pick up any signals of the DNA base pairs that it's trying to read. This is because I found out later that the DNA OLIG2 that I put in was mouse, and the primers that I had used to induce the mutations were human. So this mismatch in species of DNA ultimately caused this bad sequencing and nothing to be made. In the second trial that I conducted, the sequence was great because I did use the right primers this time, but the tyrosine, the P-tag drop-off site that I was trying to mutate, remained unmutated. So this is the normal version of OLIG2. That's not really what I wanted, because I wanted these mutant versions so that I could conduct future experiments with them. So what are some next steps in my journey to attempt to create mutant versions of OLIG2? So instead of taking the original gene and actually going in and trying to X out those tyrosine sites, I am currently in the process of completely synthesizing the gene from scratch. And all of this in hopes of getting that mutant OLIG2 version so that I can conduct experiments against control cells to see how fin kinase and OLIG2 interact within the cells, to see if the fin kinase B can drop off these P-tag pollens onto OLIG2, and this changes the overall function of rat cells in GBM tumors. Thank you.
All right, Kathleen, we're going to ask you a few questions. We'll start with Mr. Martin, move to Mr. Molk, and then I'll ask the final ones, okay?
So how did you pick out this specific topic with OLIG 2 and the associated genetic issues?
The lab that I ended up working at focuses on only OLIG2, but I was introduced to fin kinase as like a potential interactor with it that can cause these mutations. So I ended up reading about previous research on these two proteins and then trying to design a way to test their interaction myself. And what I arrived at was this mutagenesis process of the DNA that I eventually spoke to you about.
You stated that you are changing genetic code in order for cells to be interactive, specifically with amino acids, correct? Did you encounter any trials with this in your literature review with things that are uncontrollable congenital diseases such as ALS and multiple sclerosis and muscular dystrophy? If you did not encounter what did that say? If you did not encounter this, do you see this as promising to research in these unchangeable congenital disorders?
That's a really great question. The mutagenesis process of mutating certain things to see if those things are important in like normal cells, for normal cells to do their job, is actually pretty established throughout all of these molecular biology fields. So usually the very first experiment that people do before they move on to future experiments in cells, in mice, and clinical trials of drugs is to mutate like the possible molecular thing that they are interested in or think are causing these malignant things to happen, and then see like how the cell's behaviors change. So I think this is pretty common across all fields. The only thing is that this mutation process doesn't always work in every case because it's kind of like an artificial process. So it's kind of a hit or miss depending on what gene you're trying to mutate.
All right, Kathleen, can you expand on what the successful completion of your experiment would ultimately lead to in terms of real-world applications and understanding in the medical field?
So if we were able to observe what the actual impact of fin kinase on OLIG2 is, we would just have another piece of the puzzle that tells us how these extremely aggressive tumor cells work. So it's just like one step of finding another possible alternative therapy for GBM. It's like a very beginning, like basic biology kind of observational step, but I think that it's important to continue to try to understand the molecular bases of these tumor cells' growth and spread.
Okay, my last question is, at the beginning you kind of described this genetic trick, right? And you say what you're doing is supposed to take the place of this genetic trick in terms of understanding how to deal with these tumor cells, is that right?
Well, the genetic trick was actually part of previous research
Okay.
that shows that OLIG2 is important to these tumor cells. So I'm basically kind of trying to do a similar genetic trick, only with fin kinase involved this time.
Okay, so can you maybe then expound on what the genetic trick is, excuse me, and then how what you're trying to do relates to that trick?
Yeah, so there are many ways of getting rid of a gene within a cell to show that it actually is important to a cell. So in that one, the scientists who did this experiment actually worked at like the Dana-Farber Institute in Boston, and what they did was just take a double-stranded RNA and kind of insert it into the cell so that it broke down the actual mRNA that codes for OLIG2 protein. So that's one way of getting rid of OLIG2, to say that yes, it is necessary for these cell survival, because when it's gone, they die. So what I did on the other hand, when I was researching fin, so this was like a paper that I read that kind of gave me an idea of how scientists do these things, how they find that molecules are important. What I did, instead of actually knocking down the fin gene, I went in and did a very specific and kind of more specialized method of just mutating those 3 sites on OLIG2. Instead of getting rid of the whole gene, I only got rid of those 3 specific places on the protein because I knew that's where fin would be dropping off its p tags.
All right, thank you.